Hello and welcome everybody to my channel Biotechnology Entrance, and today we will be discussing Pondicherry University MSc Entrance exam question paper of biotechnology subject. So it was conducted in the year 2013. Coming to the first question, which of the following methods for studying loss of gene function does not involve any modification of the genome? So a gene knockout by homologous recombination, RNA interference by injection of double-stranded DNA, expression of an integrated antisense transgene, all of the above. So answer is option B, RNA interference by injection of double-stranded DNA. It does not involve any modification of the genome. Then restriction fragment length polymorphism is, or RFLP as it is called, the difference in the restriction maps between two individuals of one species. Answer is option B. A reporter gene allows the gene expression to be readily measured. So answer is option B. Then two double-stranded DNA samples that are identical with respect to the number of base pairs but differ significantly in their GC content can be separated by. This can be separated by density gradient centrifugation. Density gradient centrifugation is based on the principle that particles in a solution are separated on the basis of their densities. The largest class of particle form a pellet and settle at the bottom of the centrifuge tube, leaving the smaller sized structures within the supernatant. So the two DNA samples, they differ in their GC content. So the one with more GC content, it will settle at the bottom, and the one having lesser GC content will be found in the supernatant. So answer is option C. Then pyrosequencing derives its name from the fact that pyrosequencing is a method of DNA sequencing that detects light emitted during the sequential addition of nucleotides during the synthesis of a complementary strand of DNA. So answer is option B. It detects pyrophos released during base incorporation. Then aroma in rice is due to the presence of 2-acetyl-1-pyrroline. Answer is option A. Then for glycoproteins, most commonly used probe is, the commonly used probe is lectin. It will bind to the carbohydrate moiety. So answer is option D. Phage M13 vectors are widely used for. They are used for obtaining single-stranded copies of cloned DNA suitable for DNA sequencing. So phage M13 vectors, they will give you single-stranded copies of cloned DNA. Then vectors designed to replicate in cell of two different species are called as shuttle vectors. Answer is option D. Then next one. Injection of DNA into developing inflorescence using hypodermic syringe is called. It is called as microinjection. Answer is option D. Then protein binding regions of DNA are identified by one of the following techniques. You can use DNA footprinting for identifying protein bound regions of DNA. Then a major advantage of monoclonal antibodies compared to polyclonal antibodies is that monoclonal antibodies, they have identical binding sites that recognize a specific epitope. So answer is option B, that is identical binding site and they recognize a specific epitope. So monoclonal antibodies have identical binding sites that recognize a specific epitope. Polyclonal antibodies will have many binding sites. Then next one. In which of the following organism mRNA has introns? So it is present in Chlamydomonas. It is a green alga and eukaryotic. So answer is option C. Colchicine is an alkaloid used to double the number of chromosomes. It is obtained from the corms of Colicum autumnale. Answer is option B. Then Jacob and Monod contribution related to regulation of gene expression. Answer is option C. Then OKT3 antibody is used in. I have discussed this already in the previous paper. You can go through the previous videos to get an explanation for OKT3 antibody. It is an immunosuppressant. Zinc fingers are important in cellular recognition because they are structural motifs in many DNA binding proteins. So answer is option C. Then which of the following genera fix nitrogen non-symbiotically? Answer is option D, Azotobacter. Then methionine and dash are having single codon. Methionine and tryptophan, they have only single codons. So they are coded by only a single codon. Answer is B. Then D-glucose and beta-D-glucose are referred to as anomers because they differ only in the position of this particular OH group at carbon 1. That is anomeric carbon at carbon. So the alpha-D-glucose and beta-D-glucose have 5 chiral carbons, but since they have no plane of symmetry, they are not epimers. But the alpha-D-glucose and beta-D-glucose are anomers as they differ at carbon 1 only. See, the position of OH is on the right side in case of alpha-D-glucose, whereas it is on the left side in the case of beta-D-glucose. So answer is anomers. Option C. Then next one. The main rate limiting enzyme in heme synthesis is. So the main rate limit is delta ALA synthetase. That is the first enzyme in the pathway. Aminolevulinate synthase catalyzes the condensation reaction of glycine and succinyl-CoA to form 5-aminolevulinic acid. This condensation reaction takes place in mitochondria and is the rate limiting reaction in heme biosynthesis under most conditions. So ALA is aminolevulinic acid, delta ALA synthetase. Then which drug is referred to as wonder drug? It is penicillin. This is also a repeated question. Parkinson's disease is associated with underproduction of dopamine. This has also been a repeated question. I think it was repeated last year. If you see to that video, then you will get an explanation for this. Then next one. The inducer. Inducer combines with the repressor and prevent it from binding to the operator. Answer is option B. The inducer is lactose in lac operon. Creation of dolly is a phenomenon of pluripotency. This also I have discussed in one of the previous question paper. This is a repeated question, and if you refer my previous videos, you will get an explanation for this. Then identify the following point mutation in mRNA. This is also repeated, I think, third time. So answer is frame shift and transition. If you refer my previous question papers, you will get the explanation for this. Answer is frame shift and transition respectively. So option D also they have given as a printing mistake, frame shift and transition. So option D is also correct. Then integral membrane proteins are helped to locate across the lipid bilayer by using an alpha helix made up of amino acids with hydrophobic side chains. So integral membrane proteins are helped to locate across lipid bilayer. Lipid bilayer is hydrophobic. So if the amino acids have hydrophobic side chains, then they can easily be located in the lipid bilayer. So using alpha helix made of amino acids with hydrophobic side chains is the answer. Somatic gene therapy attempts to correct a gene defect by introducing the normal gene into the cells in the patient's body other than egg and sperm. So that is somatic gene therapy. The enzyme following Michaelis-Menten kinetics show a characteristic graph when substrate concentration is plotted against velocity. The nature of this graph is it is hyperbolic. Answer is option C. What is the major protein responsible for 75 to 80% of the osmotic pressure of human plasma? So it is albumin that is responsible. So answer is option C. Then next one. Which anti-cancerous drug is obtained from Catranthus roseus? Vincristine and vinblastine. They are produced as anti-cancerous drug by Catranthus roseus. In the option, vincristine is given. So answer is option A, vincristine. This is also a repeated question. Then, rhodopsin is a transmembrane protein belonging to the large family of G protein coupled receptors. It is found in the disc of rod cell of human retina. Activation of rhodopsin is due to. So, rhodopsin is activated by the photon triggered isomerization of retinal in the ligand binding pocket. Rhodopsin consists of the protein opsin linked to the 11-cis retinal, a prosthetic group. So, photoisomerization of its prosthetic group is the correct answer. The next one. The mushroom poison amanitin is an inhibitor of the synthesis of, mRNA. Amanitin is a cyclic octapeptide which inhibits RNA polymerase II, thus interfering with protein synthesis. And RNA polymerase II, what it does, it catalyzes the synthesis of mRNA. So, mushroom poisoning with amanitin will lead to the inhibition of synthesis of mRNA. Then next one. HeLa cells were obtained from, they were obtained from Henrietta Lacks. So, answer is option B. An example, all of the following cell types contain the enzyme telomerase, which protects the length of the telomeres at the end of the chromosome except. So, telomerase is not usually active in most somatic cells, that is cells of the normal cells of the body, but it's active in germ cells, the cells that make sperm and egg, and some adult stem cells. And telomerase is active in 85 to 95% of the cancers. So, answer is option D, somatic. Next one. What is the function of initiation factor IF3? It is option D. If bound to the 30S subunit, it prevents the association of 30S and 50S subunits. So, answer is option B. Short 200 to 500 base pair DNA sequence that has a single occurrence in the human genome and whose location and base sequence are known as. So, express sequence tags are fragments of mRNA sequences delivered through single sequencing reactions performed on randomly selected clones from cDNA libraries. So, the answer here is sequence tagged sites. So, short DNA sequence that has single occurrence in the human genome. So, they are sequence tagged sites. Then, the vitamin is the major lipid soluble antioxidant in cell membranes and plasma lipoproteins. That is vitamin E. Answer is option D. Amphibian metamorphosis is controlled by which hormone? That is thyroid hormone, option A. Which of the following drives directional transport through nuclear pore complexes? So that is done by RAN GT pace. Then positional cloning refers to, it refers to mapping a gene to the chromosomal region and then identifying and cloning the genomic copy of the gene from the region. So answer is option D. Then next one. The mitochondria serve as a marker for cytochrome oxidase and the lysosome serve for acid phosphatase. Answer is option B. Then lysosomes are abundant in WBC and spleen. Answer is option D. Then which of the following statements about transmission electron microscopy is not true? The specimen must be stained with osmium or other heavy metal. Then the specimen is placed in a high vacuum for viewing. The specimen must be sliced very thin to 20 to 100 nanometer in thickness. The beam is focused by electromagnetic lenses. So the answer is option A. Next one. Calcium absorption is inferred by. It is inferred by fatty acids. So answer is option A. Then Bohr effect is the decrease in oxygen affinity of hemoglobin in response to decreased blood pH. And how does the blood pH decrease? By increasing the carbon dioxide concentration in the blood. So the hemoglobin dissociation curve, it shifts to the right. So answer is option A. The mechanism of synthesis of DNA and RNA are similar to all the following ways except they involve release of pyrophosphate from each nucleotide added. That is correct. They require activated nucleotide precursors and Mg2+. Yes. The direction of synthesis is 5' to 3'. They require a primer. Okay, the DNA polymerase require a primer whereas RNA doesn't. So answer is option D. So next one. Albinism is due to deficiency of the enzyme. It is deficiency of the enzyme tyrosinase. So it is a copper containing enzyme involved in the production of melanin. So answer is option B. Molecular weight of heterogeneous nuclear RNA, hnRNA is more than 10 raised to 7. The T5C arm in an tRNA molecule possesses the sequence. It possesses answer A, that is T pseudouridine and C. T is thymidine and C is cytidine. So answer is option A. A synthetic nucleotide analog used in the chemotherapy of cancer and viral infection is arabinosyl cytosine. Answer is option A. Then the most likely lethal mutation is it is insertion of one nucleotide. Answer is option B. Then regulation of heme synthesis occurs by repression, derepression. Answer is option B. An inherited or acquired renal tubular defect in the reabsorption of phosphate is characterized with a low blood phosphorus with elevated alkaline phosphate. So answer is option C. The immunoglobulin which provides highest antiviral activity is. So answer is option C, IgA. The next one. Human growth hormone has one polypeptide chain and one intrachain disulfide bond. Answer is option A. Acromegaly results in all of the following except increased stature. Acromegaly is a hormonal disorder that develops when your pituitary gland produces too much growth hormone during adulthood. So increased stature is not present in case of acromegaly. Then the blood sugar raising action of the hormone of supraelevation suprarenal cortex is due to? It is due to glyconeogenesis. Answer is option A. Glyconeogenesis is a shunt for the synthesis of sugars such as glucose and glycogen from substances other than sugars. An example is the conversion from lactic acid to glucose. Then suppressor mutation occurs in the anticodons of tRNA. Suppressor mutation is a mutation in the gene for tRNA molecule that changes its anticodon loop, or else it can suppress nonsense mutation that occur in the protein coding gene. Now suppose there is a nonsense mutation occurring, that is the that result in termination of polypeptide prematurely. So that can be suppressed if you mutate, if the tRNA molecule is mutated at the anticodon loop. Then, trials for gene therapy in the human beings were first carried out with considerable success in a genetic disease called adenosine deaminase deficiency. So, William French Anderson and Michael Baez and Ken Culver performed the first successful gene therapy on a human in 1990. They developed a protocol for treating adenosine deaminase deficiency, severe combined immune deficiency, also known as the boy in the bubble disease. So, next one. If DNA of a cancer cell is introduced into a normal cell, the recipient cell, it changes into cancer cell. Answer is option D. Then next one. Amplification of dihydrofolate reductase gene in cancer cell makes the cell resistant to amyotropin therapy. So answer is option C. Then orsinol method is employed in the quantification of RNA. So answer is option C. Which of the following statement is not characteristic of allosteric enzymes? Allosteric enzymes, they follow Michaelis-Menten kinetics. So that is the answer. But actually the allosteric enzymes, they do not obey Michaelis-Menten kinetics, but instead they have sigmoidal kinetics. Then clearing factor is a lipoprotein lipase, and it is responsible for the removal of most of the plasma triglycerides from the bloodstream. Then next one. Rapport Lubbering cycle is located in the erythrocytes. In this cycle, 1,3-biphosphoglycerate is converted to 2,3-bisphosphoglycerate by an enzyme bisphosphoglycerate mutase. 2,3-bisphosphoglycerate is then converted into 3-phosphoglycerate by enzyme 2,3-bisphosphoglycerate phosphatase. Then what is the significance of this cycle? The 2,3-bisphosphoglycerate combines with hemoglobin and reduces the affinity towards oxygen. So in its presence, as oxyhemoglobin unloads more oxygen to the tissues. Then 2,3-bisphosphoglycerate, it shifts the oxygen dissociation curve to the right. So coming to the next question, in Lien-Weaver-Burke plot, the y-intercept represent. So in Lien-Weaver-Burke plot was widely used to determine important terms in enzyme kinetics, that is Vmax and Km. The y-intercept of such a graph is equivalent to the inverse of Vmax or 1 by Vmax, and the x-intercept of the graph represent minus 1 by Km. So answer is option A, Vmax. Genetically engineered male sterile crop plants may be produced by inserting Barnase gene. So chimeric ribonuclease gene or Barnase gene expression within the anther selectively destroys the target tapetal cell layer that surrounds the pollen sac and prevents pollen formation and leads to male sterility. Now choose the correct statements about thermostable DNA polymerase used in PCR. So they have given different polymerases that are used in PCR, and we have to choose the correct answer or correct statement about these thermostable DNA polymerases. So TLI pol has no 3' 5' exonuclease activity. It has 3' 5' exonuclease activity. Then PFU pol has 3' 5' exonuclease activity. That is correct. That is proofreading activity. That is 3' 5' exonuclease activity, meaning that as the DNA is assembled from the 5' end to 3' end, the exonuclease activity immediately remove nucleotides misincorporated at the 3' end of the growing DNA strand. So Q is correct. Then TAC pol has 3' to 5' exonuclease activity. TAC has 5' to 3' nuclease activity, but it has no 3' to 5' exonuclease activity. So R is also not correct. Then TAC pol has no proofreading ability. That is also correct. So answer is Q and S are correct, but R and P are wrong. So that is not given in the options here. There is a printing mistake. So coming to the next question. Which of the following would not be possible to address using northern blots? Northern blots allow mRNA transcript size comparison with RNA size standards. So, and option A is possible with case of northern blot. Then location of restriction sites in a particular gene, that is not possible with case of northern blots. And spatial regulation of a particular gene and temporal expression of a particular gene, that is also possible with northern blot. So what is temporal regulation or expression? A type of regulation of gene expression in which a gene is only expressed at a specific time in the development. For example, genes that are only expressed in the light or during flower development. Then spatial regulation, a type of regulation of gene expression in which a gene is only expressed in a specific location in the plant, that is seed storage proteins. So answer is option B, location of restriction sites in a particular gene. Then in lac OC, OC is constitutive operator. So the operator will keep on synthesizing the protein product whether the repressor is present or not. Is a partial diploid of the two LAC-Z enzymes. Only the mutant enzyme LAC-Z minus is synthesized constitutively. This observation shows that LAC-OC mutation is a cis-dominant. Cis-dominant is the operator, that is OC operator, controls only genes on the same DNA strand. So it is a cis operator gene. So answer is option A. Now chromosome walking is described as. Answer is option B, identifying clones whose inserts overlap to generate a library of clones that cover a given segment of DNA. Now next one. A mixture containing two proteins having similar molecular mass but different oligomeric properties can be separated by. So they can be separated by native page analysis. Native page separates proteins based upon their charge and mass. Because non-denaturants are used in native page, subunit interaction within a multimeric protein are generally retained and information can be gained about the quaternary structure. So a mixture containing two proteins having similar molecular mass but different oligomeric properties. So they have similar molecular mass but different oligomeric properties, and they can be separated easily by a native page analysis. Then next one. Which of the following antibiotics attaches to 50S ribosome and inhibits peptidyltransferase activity? So answer is chloramphenicol. Option D. Trimethoprim binds to dihydrofolate reductase and inhibits the reduction of dihydrofolic acid to tetrahydrofolic acid. Now this THF, that is tetrahydrofluoric acid, is essential precursor in the thymidine synthesis pathway, and in interference with pathway inhibits bacterial DNA synthesis. Then amphotericin B is an example of polyene type of antifungal. Then polyene binds to fungal ergosterol, that is the primary sterol in the fungal cell membranes, and this alters cell membrane permeability and intracellular components leak from the cell. Now next is penicillin. Penicillin kills susceptible bacteria by specifically inhibiting the transpeptidase that catalyzes the final step in cell wall biosynthesis, the cross-linking of peptidoglycan. So answer is option D, chloramphenicol. The technique used to identify specific DNA sequence in bacterial colonies is, so answer is dot blot technique. It is colony hybridization technique. It is colony hybridization technique. So in colony hybridization technique, in genetic lab technique used to identify which colonies of bacteria on an agar plate contain a particular sequence of DNA or a particular gene. Then next one, RFLP involves. RFLP is used to identify a specific DNA. So answer is option A, restriction fragment length polymorphism, that result from variation in DNA sequence recognized by restriction enzymes. So it is used to identify specific DNA. Then which of the following are vectors for animals? The answer is option C, SV40 vectors and bovine papilloma virus vectors. Then next one, which of the following chemical enhances Vir gene expression? So the answer is option A, acetosyringon. Then RNA is very much susceptible to hydrolysis in alkali because its 2' OH group participates in intramolecular cleavage of phosphodiester backbone. So answer is option B. Then next one, which of the following fluorescent probe is used to monitor the progress of amplification of real-time PCR? Answer is option A, cyber green. Drugs that either stabilize or depolymerize microtubules can be used in cancer chemotherapy. Which of the following is correct concerning such drugs? So they interfere with mitosis. Answer is option A. The rate limiting step of fatty acid synthesis is catalyzed by, this is also a repeated question. It is the formation of malonyl CoA from acetyl CoA. That is the rate limiting step in fatty acid synthesis, and it is catalyzed by the enzyme acetyl CoA carboxylase. Answer is option A. Then delay of senescence due to cytokinin is known as Richmond-Lang effect. Cytokinins retard aging in plants. The effect is called as Richmond-Lang effect. Richmond-Lang effect is retardation of aging due to cytokinins, which acts by retarding the rate of destruction of chlorophyll and yellowing of leaves. Next one. A biochemical indication of vitamin B12 deficiency can be obtained by measuring the urinary excretion of methylmalonic acid. Answer is option C. Fully activated pyruvate carboxylase depends upon the presence of, it depends on acetyl CoA and biotin. Answer is option B. Stain which is not useful in identifying fungus. It is option D, PAS, periodic acid Schiff. That is not useful for identifying fungus. Then COT unit is rate of renaturation per second. Answer is option A. Then when bacteria produce mammalian proteins, cDNA is used rather than genomic DNA. Which of the following is best explanation? The most eukaryotic genes have introns that cannot be removed in bacteria. So when bacteria produce mammalian proteins, cDNA is used rather than genomic DNA because there are no proteins which will cleave off the introns in bacteria. Then next one, which cycline protein involves at late G1 phase of the cell cycle? Answer is option B, that is E cycline. E. Next one. A researcher performs a cross between two mice, both having black fur. Black fur dominant over white. White fur. 75% of the offspring have black coats and 25% have white coats. The researcher can assume that the parent's genotype is more likely. It is capital B small b into capital B small b. So you will get these gametes, and if you cross you will get 3/4, that is 75% black and 25% white. So answer is option D, capital B small b into capital B small b. Then next one. In mammalian cells, rRNA is produced mainly in the nucleolus. Then caffeine promotes lipolysis by increasing cyclic AMP levels through its effect on the activity of the enzyme phosphodiesterase. Answer is option C. Then rice bacterial blight resistant gene XA21. Answer is option A. Then isoelectric point of alanine is 6. If alanine is dissolved in a buffer of pH 3 and subjected to electrophoresis, so what will happen? At pH is equal to Pi, the amino acid is Witter ionic and carries no net charge. This is called isoelectric point of the amino acid. At this pH value, the amino acid will be stationary in an electric field, that is pH is equal to Pi. Now what will happen at low pH, that is pH is less than Pi, as is the case given here, that is 3 is less than 6. The amino acid carries positive charge and it will migrate towards the cathode. So answer is option B, it will migrate to the cathode. Then consider the following DNA sequence. Due to frameshift mutation, there is insertion of G between 3rd and 4th position. Consider a reverse mutation occur in the same mutated sequence. Which reverse mutation will have minimum effect in protein change? So out of these options, which one will have minimum effect in protein change? That is, most of the protein amino acid sequence are retained as in the case of the original parent. So in which one the reverse mutation will have minimum effect in protein? It in the next slide. So here this is the sequence that they have given. So option and I have separated into 3 nucleotides for easy understanding. Then option A, they have given insertion between 5th and 6th. So 1, 2, 3, 4, 5 and 6. So over here I am written the introduction, that is insertion of the nucleotide as X. So after insertion you will get the sequence ATG, GGX, CCA, TAG, ACG, ATA, TGG and TAG. Now if you insert between position 5 and 6, then you are not going to get a protein which will code for the same amino acid as the wild type. So insertion between 5 and 6 is not the correct answer. Now if you deletion between 5th and 6th position, Now if you delete at 5th and 6th position, that is this C will be deleted. So you will get ATG, GGC, ATA, GAC, GAT. So this is also not same as that of this particular amino acid sequence. So this, then next one, insertion of 3 nucleotides between 5th and 6th position. So between 5th and 6th, that is over here itself, you are inserting 3 amino acids or 3 nucleotides. So I have given 3X over there. Now if you insert, you will get ATG, GGX. X can be any base, and here you will get XXC, CAT. So it is resembling CAT over here. AGA, AGA, CGA, CGA, TAT, TAT, GGT, GGT, AG, AG. So insertion of 3 nucleotides doesn't cause much variation in the protein sequence. So answer will be option C, that is insertion of 3 nucleotides between 5th and 6th position. Next, option D, if you see deletion of nucleotide between 11th and 12th position. So over here if you delete, this half of the protein will code for, half of this particular DNA sequence will code for the same protein as that of the wild type, but the other half will not have the same amino acid sequence as the wild type. So answer is option C. Then mucus which covers the epithelium lining of stomach protect it from protease activity is secreted by. So it is secreted by columnar epithelial cells. So that is not given in the options. So mucus covering the epithelial lining of stomach is secreted by columnar epithelial cells. Then next one, the largest protein in the body is titin. Answer is option C. Which of the following DNA replication or repair system is dysfunctional in individual with xeroderma pigmentosum? So it is nucleotide excision repair. Xeroderma pigmentosum is a rare human autosomally inherited skin and neurodegenerative disease that is associated with very high incidence of skin and mucous membrane cancers due to exposure to UV radiations. So that is xeroderma pigmentosum. Then on a Ramachandran plot, the entries for hemoglobin would be clustered around. They will be clustered around the right-handed alpha helix. Answer is option C. So that's it. Thank you for watching. Please like, share and subscribe. Thank you.